Hello everyone, today we are going to start the hallmarks of the cancer. The cancer is caused by the multi-sequence DNA mutation that affect the fundamental cellular processes like growth, DNA repair, and survival. Due to these features, we can differentiate the cancer cell within normal cells. And they show some hallmarks and features, and we are going to talk about them one by one. The first is a growth signal self-sufficiency. It is a hallmark of a cancer refers to the ability of cancer cells to proliferate independently of external growth signals. The normal cells require signals from outside to grow and divide, but the cancer cells then become autonomous, generating their own signals or ignoring the external cues when inhibitors. And this will happen due to mutations in genes encoding. It can be due to protein cogenes that is responsible for the cell proliferation that increases cell growth. And due to overexpression of protein cogenes, there will be increase in growth of dead cells. Taking the growth factor receptors. Growth factor receptors also constitute for the signal most likely HER2 in the breast cancers. These are signaling molecules. Here's some signaling molecules like the ROS proteins. These are the small guanosine triphosphates. You can also say GTP-6 that act as molecular switches regulating cell growth, differentiation, and apoptosis. And due to that, these are colorectal cancer, lung cancer, and pancreatic cancer. And transcription factors like MIC, MSC gene, two types like MSC and MIC. And okay, there are two types, MSC and N. The MSC causes the bulkhead lymphoma, and MSCN causes a neuroblastoma. Okay, and this is said cyclogluteus. Said cyclogluteus also responsible for said proliferation in diva cyclins and cyclin-dependent kinases. Cyclin-dependent kinases and the other key regulator of the cell cycle, ensuring its progression through various phases and maintaining genomic stability. Actually, these cyclins activate CDKs, which then phosphorylate and target the proteins and driving a cell forwardness cycle. And these CDKs are the family of kinases that are inactive until they attach with cyclin. The cyclin-dependent kinases are inactive, but it will become active when they are attached to cyclin. The second hallmark is anti-growth signal insensitivity. The anti-growth signal insensitivity mutation in tumor suppressor gene. Anti-growth signal insensitivity is basically mutation in tumor suppressor cells, tumor suppressor genes. What is happening if you guys saw more than G1 phase and cell cycle and the S phase? In both phases, it's a replication phase for the DNA. It is in this phase is a replication of DNA. Between this point is a checkpoint that is controlled by RB proteins. These proteins are responsible for the checking the cells are they are okay and they are proper and they are having no defect, there is no abnormality cell to divide and it is not cancerous cell. The checkpoint is controlled by RB proteins. If there is a defect or inactivation of RB proteins, then there will be mutations and there will be cancerous cell growth. Because this is a checkpoint now, so many of cells are abnormal, did not able to grow, but we are sending them to elsewhere that will become dangerous, and in the future they will become cancer cells. And we'll see how these will be active. RB is active, but after phosphorylation of RB become inactive. The phosphorylation of RB causes inactivation of RB proteins. The next thing for the anti-growth signal insensitivity first, tumor suppressor gene. If tumor suppressor genes are inactive, then there's no Other thing that can stop the growth of cancer, taking loss of E-cadherin function. The E-cadherin is a calcium and calcium-dependent transmembrane glycoproteins that play a crucial role in cell adhesions, particularly in epithelial cells. And it also acts as a tumor suppressor and by inhibiting the VNT signaling pathway. Inhibiting the VNT signaling pathway, and also to be responsible for the cell contact of cells. Like two cells are attached with each other, and it's mostly responsible thing is E-cadherins. Okay? And when the E-cadherins lose their function and leaves lots of contact inhibition due to NF2 mutation, there are many of cells getting separating. So if one cell is in a group, one cancer is in a group with some cells, then E-cadherin lost their function, then these cells is separate to different places. Our next hallmark is evasion of apoptosis. Like inhibition of apoptosis. In the apoptosis, we already know there is some pathways, extrinsic and intrinsic pathways. Okay, you guys know there's a BCL2 expression or a liking. If BCL2 expression is increasing, there's no release of cytochrome C. Cytochrome Cs don't release in there living no apoptosis. Same happening in mutation in their genes, they regulate the apoptosis and due to regulate the BCL2 and TP53 defect, and it will result in follicular B-cell lymphoma. You divide, the cell will not go to dead process and it will be start growing, and it will result in the cancer. Then the limits the duplicate to potential. Actually, these cells are retuition of telomeres. Telomeres. Actually, telomeres are responsible for the maintenance and maintaining of telomeres is beneficial for the cell, for the dividing. Like, as longer the telomeres is, the cell will divide as much. Okay? The shortening of telomeres in unable to divide, the shortening of telomere did not able the cell to divide more. So, it is just like a aging. Cell aging, the cell aging can be detected by the telomeres, but in the cancer, there's a reactivation of telomere to divide. There's a prevention of chromosome shortening and cell aging. So, always the cell can still become young and it will divide and divide as much it can. So, the females are using botulinum and Botox for the anti-aging process, but the cancer cells are using telomerase reactivation to prevention of chromosome shortening and cell aging. Understand? Next is sustained angiogenesis. Lex cancer cells also want some nutrition, oxygen for survival through they make new blood vessels. They survive, for their survival they make new blood vessels, and that process is called angiogenesis. And for that the responsible factors are VGF and FGF, the fibroblast factor. The both factors are important factors for the angiogenesis, and they are responsible to divide new blood vessels will form, and cancer cell will take their happy meals like nutrition, oxygen, and the thing they want. Okay? And these vessels can be separated from existing capillaries like new angiogenesis, where endothelial cells are recreated from the bone marrow and with the vasculogenesis. The vessel may be leaky or dilated. It depends on the feature of the cancers. But the angiogenesis, a more important point, what an important factor that VEGF and FGF. I know a third factor either, Wahlberg effect, the Wahlberg effect, is that taking of glucose with the cancer cell. How much glucose is cancer cell taking? Okay? And this is a shift to glucose metabolism away from mitochondrial oxidative phosphorylation toward the glycolysis. It is a property for cancer cell is take high quantity of glucose from the metabolism. Okay? And for generating high number of ATP. For that, and we are using this process. we are using this thing in PET scan. In PET scan, but what we are doing, we are giving radio-labeled radio-labeled glucose. When you give the radio-labeled glucose, the abnormal cell will take high amount of glucose of that. And that will be shown on PET scan by a shiny colored slit, okay? Again, it's a very important point for the PET scan, which effect is using the Wolver effect. The Wolver effect is taking of glucose and how much glucose you consider taking, and by PET scan we can detect the consistencies by giving that radio-labeled glucose. And they are saying aerobic glycolytes provide rapidly degrading cancer with the carbon-native synthesis of cell structure and it leading to increasing lactic acids. Taking immune evasion in cancers, actually immune cells also detect cancers and destroy them. Okay? It is not like that cancers are independently, but the immune cells having the capability to destroy the cancer cells, but they have to do something for their survival. At this we are also doing for our survival many things we are doing. Like in winters we are taking the jackets. In summers we are wearing some cotton clothes like that. Seem like that the cancer cells are making them of decisions to fight against the immune evasion or escaping from their attack. For example, someone found to hit you, you are not a capable person, you are not fine to fight against them, then you have to escape. Seems like the cancers are doing and they are doing some escaping system, how they are taking the modification we are read now. Normally that means they can recognize and attack the tumor cell. For successful tumorigenesis, the tumor cell moves evade the immune system. Multiple escape mechanisms exist. The first one it decreases molecular histocompatibility expression. If the tumor cell is now showing MSC class 1, then tumor and then the T cell did not recognize them. Due to unable to detect MSC class 1. And due to this expression, lack of this expression, they did not recognize tumor cell and T becomes safe. And cytotoxic T cell unable to recognize the tumor cells. It's a simple thing. If someone is showing MSC class 1, the T cell will detect them and destroy them. But when the cancer cell is not showing decreased expression of MSC, the T cell did not recognize them, unable to recognize, and they will not destroy them. And second thing is, tumor cells secrete immunosuppressive factors, that tumor growth factor beta and regulate T cell to do regulate the immune response. These are some responses that are anti-inflammatory response due to which automatically your immune system did not respond well and they will think, okay, chill, everything is normal in the body. But the third thing, the tumor cell regulate immune checkpoint molecules which inhibit the immune response. This is an important thing. First, you have to know what are immune checkpoint molecules. Immune checkpoint molecules are proteins that regulate the immune system, preventing it from attacking normal cells and tissues. And they act as a brake in your immune system, ensuring that immune responses are balanced and don't cause excessive damage. How do tumor cells exploit immune checkpoints? Okay, the tumor cell can take advantage of these immune checkpoints to evade the immune system. By upregulating increased expression of certain immune checkpoint molecules, tumor cell can send inhibitory signal to immune cells, such as T cell, that would otherwise recognize and attack the tumor. The key players for that, for inhibiting the cells are PD-1. PD-1 this is called a proground, that one, a protein on T cell, and is a prisoner, T cell. Okay? PD-1 is a proground. That one. And these are present on T cell and That when bounded ligand PTL1, the T cell activation and function inhibits. For inhibiting the T cell, these are one ligand on the T cell that a PD1 programmed that one when it attached with the PDL1. PDL1 is a program that ligand one. This causes inactivation of T cell, okay? And the third molecule is CTLA form. It is a cytotoxic T lymphocyte antigen, cytotoxic T lymphocyte antigen, and this is another protein that regulates T cell activation and can be targeted by tumors to evade the immune responses. Don't forget it's an important thing, it's also present in another topic like PT1, PDL1. The PT1 is present on T cells. It will attach with PDL1 and become inactive, and there's one more protein, antigen CTLA4. Okay? Now we are doing the next part at the tissue ingredient. The tissue ingredient is due to lose ecadine function road ID. No, the cell contacted due to ecadine when the ecan helilosis function, given the losing of intracellular junction and the metalloproteinase, declare the basement membrane and ECM. What's ECM? The ECM is the extracellular matrices. These proteins are crucial suction signaling component found in a space around cells and in tissues. They provide us support, facilitate the adhesions, and influence the cell behavior through interaction with cell surface receptors. And due to these, give it also metalloproteinase enzymes action like collagenase, hydrolysis. This all enzyme will act on basement membrane and it will disrupt it to develop cells attached to ECM proteins like lamin, fibronectin, and cells migrate through degraded ECM and then the ribovascular dissemination. Okay. And don't forget the ECM protein. The ECM proteins includes collagen, elastin, fibronectin, laminin, proteoglycans, and other also like osteoportin and fibrinogens. These all proteins are ECM proteins, and it is happening in vegan how? First, ecadine will lose its function after there will be metallo. Proteinase enzymes act on basement membrane after the disruption. The cell will start invasion and start dissemination in vascular system. Understand? Now we move to the last hallmark of cell consistent, that is metastasis. What is happening in the metastasis? In metastasis, the cell is going to separate to other organs, and tumor cells will embrace separate with a lymphatic supply. It happens at the end we don't hit him, and that's an extra variant and the harming. The site of metastasis can be predicted by the site of primary tumor, as a target of being often the first encounter capillary blood. Some cancers show organ tropism, but the organ tropism means the tendency of the cancer cell to the perfectly separate to specific organ tissue. This means that not all organs are equally likely to harbor the metastatic tumors. Some may be favored over others. Okay? Like if there's a prostate, then it is going to want to burn white cells, right where veins. And if it is like TIT, it is going to leave our lungs like that. Okay. If any blood separate it first going to lungs like that. Every organ have their own specific first site of metastasis. And for that things, we have to be known their origin, how they are going to separate. And secondly, there is some other processes, some other important points I want to share in. Like already you guys know there are two types of separating by lymphatics or by the blood. There's some important point like sarcoma. If you guys know the sarcoma is within gynecology cancers, and these spread via blood hematogeneously. Okay, like artery thick wall, and you guys know the arteries having the thick walls, and due to which they the difficulty to penetrate. And secondly, the veins, wins the thin wall, they can easily penetrate and leave along the most common sites of hematogenous spread. So the sarcoma cancers are going to spread through blood, and if any cancer is spreading from the blood, then it most commonly go to liver or lungs. Understand? And what is our second route? The second route is lymphatics. The carcinoma. The carcinoma is separate through lymphatics. But these are exceptions. Exceptions are for carcinoma spread by bloodstreams. For the choriocarcinoma. Second is renal cell carcinoma. That is spread by renal vein, then hepatocellular. Again, hepatocellular carcinoma, that's a place by portal vein. And one is follicular. Thyroid carcinoma. Okay? Already I done with that quality. In metastasis is the important thing. The roots, there are two routes of spreading the cancer for the lymphatics and blood. The sarcoma mesenchymal cancers are spread by the blood. And whenever any cancer is spreading by blood, it will go to liver or lungs. And all the carcinomas are going to lymphatics route, but there is the exception of all cancers: choriocarcinoma, renal cell carcinoma, hepatocellular carcinoma, and follicular thyroid carcinoma. And it's all about the metastasis and hallmarks of cancers. Now we are going to start immune checkpoint interaction. Immune checkpoint interaction, we already know. It's a hallmark of a cancer like immune Evasion cancer. In that, normally immune cells can recognize and attack the immune and attack the tumor cells. For successful tumorigenesis, the tumor cells must evade the immune system. And for that, it having a multiple escape mechanisms. First one decreases the expression of molecular histocompatibility, class 1. The second, releasing the immunosuppressive factors. And turn, they are making some regulation in immune checkpoint. From this escaping mechanism, we are going to study how it is going to make the immune checkpoint regulation and how we are going to overcome this effect to treat the cancer. If you guys know, there's PD1 program, that is present T-cell. And on cancer, there's one element, like PD11, that are going to attach with PD1 and cause inactivation of T-cell. When PD1 and PD11 interact with each other, attach with each other, it causes inactivation of T-cell. And secondly, there's one more mechanism. Normally, CD28 have to attach the B7 of tumor cell to cause destruction. But here it making one escaping mechanism. The B7 will attach with CTLA4 to cause the inactivation of T cell. To prevent these escaping mechanisms, we are going to start cancer immunotherapies to increase our immune response against cancer cells. And first, already we're done with intriguing between PD1 and the PD-L2 that cause the inactivation of T-cell dysfunction. And to inhibit that port, antigens of port interactions like PD-1 and PD-L2, we are going to inhibit with some drugs. we're having seplizumab, nivolumab, pembrolizumab for inhibition of pinky vine. For other factor like PD11, the proground lichen 1, we have the menu antibodies like atezolizumab, nivolumab, and avelumab. And this is seeking traction at CTLA4 that is going to attach to P7 instead of CD28, and it will also it with the loss of function of T cell. And to stop this interaction, we are going some antibodies like epilumab. Epilumab is going to inhibit which function of CTLA4. By these immunotherapies, we are going to increase immune response of the body against the tumor cells. Okay? After the immune therapies, it will increase. We are going to start cancer epidemiology, in which data cancer incidence and mortality. There is a question, what is the incidence and what is mortality? The incidence being the rate at which the new cases of a disease or event occur within a population. In simple words, you can have frequency of cases. And mortality means the death. The state of being subject to death or the relative frequency of death in the population. Mortality means death frequency. The incidence is the frequency of new cases. Frequency of new cases. And mortality is the frequency of death after getting that disease. First, we are talking about the incidence. The incidence, the most common cancers are prostate and breast. Prostate in male and breast in females. But the second most common cancer for the new case frequencies are lungs, and third is colon and rectum. These two cancers did not discriminate the genders, but the most common cancer, like prostate and breast, is having its discrimination upon the two gender. Like prostate are most common in male and breast is most common in female. If we talk about the children are most common cancer in leukemia, the second the CNS, and third the neuroblastoma. And this day, if we're talking about the mortality to which cancer is the most common days are going to death. The most commonly lung diseases causing this interval without discriminating the gender. The means female both are going to die due to the lung cancer, the most common cancer according to mortality. The second is prostate and breast, and third is colon and rectum. The lung cancer incidence has decreased in the males, but has not changed significantly in females. The cancer is secondly leading cause of death in the United States, and heart disease is the first. In all the diseases, the heart disease is a first leading cause in USA. And cancers are second leading cause. And guys, if you talk about the skin cancer, the skin cancer also having some impact. And in that, the most common cancers are beta cell carcinoma. The second in skin cancer is squamous cell carcinoma. A real one is a melanoma, but the melanoma are so aggressive and easily spread. Its metastasis high rate as compared to squamous and beta. The beta and squamous cell carcinoma are the most common type of skin cancer, while melanoma is a more serious type that can spread. Beta cell carcinoma arises in the pubescence in the lower part of epidermis. The squamous cell carcinoma develops in squamous cells in the outer layer of skin, and melanoma originates in melanocytes with producer pigments. Okay? Again, I'm saying the PCC, basal cell carcinoma and squamous cell are less likely spread than melanoma, but all three can be treated. If all about the cancer epidemiology, kindly So bring awareness and protect yourself. Thank you.